Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class we have looked at a FASTQ data file. We have explored this with VI editor, and then we have set up the FastQC tool and we have at least run the FastQC tool. Okay. So, what we are going to do today is we will actually look at the results of that FastQC run, right. We will evaluate, right, if there is any issue with the data, if there is any any problem with the data. Okay. Once we do that, we will then do some pre-processing if required. Okay. And so, if trimming, for example, if we require trimming, we will do that. Okay. So, these are the topics that we will actually see today, right. One is data quality evaluation on the results that we have generated already, then we will do data trimming if it is required, and then finally, after we have done trimming, we need to redo the quality control. Okay. To ensure that the trimming is working fine, we have actually removed the problem that we had in the beginning. Okay. So, this is something that we will discuss. These are the steps that we will be following, right. So, if you can visualize in your mind, we have done the data quality control. So, once you do that, you have to evaluate the results, right. Whatever results you have generated, you look at the results and then make decisions if you need to do pre-processing, for example, trimming, right. So, we have talked about two different types of pre-processing, one is duplicate removal, the other is trimming. So, if this is required, for example, trimming, we will have to do the trimming, but then once we do trimming, we have to go back to the evaluation stage again, right. We have to run QC, we have to do the evaluation, and if something is still not okay, we have to again do pre-processing, right. So, this is an iterative step, right. We do the quality control, we have the evaluation, we do the pre-processing, we again do evaluation, quality evaluation. Okay. So, we will actually do this cycle today, right, with the data set that we have already seen in the last class. Okay. So, let us go there, let us move into the terminal, right, where we have actually generated the quality control. So, we are inside the terminal, right, and if you remember in the last class we have discussed how we generate these quality control results using FastQC. We have run the command, and inside these results FastQC we have moved all the results for these two files, right, read 1, read 2. Okay. So, we have extracted these results, and what we are going to do, we will actually now look into the results, we will make our evaluation, and then we will continue with pre-processing if required. Okay. So, let us go inside the folder now. Okay. Inside this folder we will see the results. Okay. So, this is results FastQC, and we have extracted this, right. Well, I will give a zoom in, right, so that it is easier, and here is the result, right, for read 1. Okay. And you will find a lot of files, but there are two files, one file that is will give you the summary, and that is the most important file, and then you will have some images folder, right. So, we will look into these two things, these are the two main things in this analysis. Okay. So, if you look at the summary, right, you will notice some statistics, right. So, here is this basic statistics, right, what you have generated by this FastQC. Then you have this T12, the name of the file that you are working with, and then on the left side you will see this pass, fail and warning. Okay. So, this is something that will tell us, right, whether the quality is okay, right, based on these statistics. Okay. So, we have talked about these statistics in the theory class, right, what kind of statistics you get and what do they mean, right. So, you have per base sequence quality, per tile sequence quality, per sequence quality scores, etc., right. All of these things we have discussed in detail what they actually refer to. Now, what is important here is that for most of the statistics, right, we see pass, right. So, they are of acceptable quality. Okay. But for two statistics here we see fail, means there probably is some sort of issue that FastQC have detected. Okay. And this is where we need to actually look into the images, okay, because those images will tell us what could be the problem. Okay. So, this fail, the first fail is for per base sequence content. Okay. Again, without looking at the image, we cannot tell what is the problem, why it is giving us this fail kind of remark. And then for the adapter content, you also have fail. Now, this one probably raises a flag, right, immediately. If you get fail for adapter content, means there is adapter contamination in the data. Okay. So, we need to probably do some sort of trimming. Okay. So, let us now go into the images folder, right, keeping this in mind that there are two statistics for which we have this fail remark. Okay. And these two statistics we have will look into and carefully, very carefully, but we can also look at the other ones. Okay. For example, we can start with this power based quality, right. We have discussed what this means and we have seen these kind of figures before. Okay. And we know what is x axis, we know what is y axis. Most of the quality score is in the green zone. So, this is acceptable. We see some low score in the beginning, but that is again expected of Illumina, right. We know why that is the case, we have discussed that, but most of the quality score is in the green zone. So, means the read data is pretty good. Okay. We can look into other statistics and this is one statistic where we have this fail remark, right. This is the sequence content across all bases, right. So, we have this position along x axis and we have percentage ATGC along y axis. Now, the fail I think comes from this part here, right. So, there is no other reason for giving this fail remark, because there is this difference in ATGC content, right. So, this is an AT biased genome. So, we have 60% AT and 40% GC. So, that is okay, right, because it will vary from one organism to another, but here in the beginning you have a lot of fluctuation, right. So, this can actually give this fail. So, this is okay again, right, considering the fact that you will get some sequencing issues, right, in the beginning when you start the Illumina run. Okay. So, we have again discussed why that is the case. So, what I will then say is that do not blindly believe all the remarks, right, pass, fail, etc. For example, here we have seen power based sequence content, it looks fine to us. Okay. We can look into the other ones, right, part sequence GC distribution. For example, we can see part sequence quality, this is an important parameter, and what we see, most of the reads are of very good quality, right. So, they are above 30 and there are very few which are below 30, but they are all above 25. This is the average quality score of the read and this looks very good. We can look into the sequence length distribution again, and sequence length distribution is 125 and that is expected for Illumina, right. Sequence length distribution will get reads of the same length. We can look into the part tile quality, right. Again it appears as fully blue, right, that is actually a good thing, right. If you remember, this is a heat colored map, right. So, if you have red or yellowish color, means there are some issues with quality score at certain regions or certain tiles, right. So, this is something that is very good. Now, the final one that we will look at is the adapter content, right. We remember, right, there was this warning, right, this fail remark against the adapter content, and you probably now understand why that is the case. So, if you look at this carefully, I will zoom in, right. X axis is you have the position in the read, y axis is the percentage of reads, right, and what percentage of reads contain adapters. Okay. So, here you see starting from, for example, around 35, 36 position, many reads start to contain adapter sequences, okay, and the color identifies which type of adapter, right. So, this color yellow means we have next era transposes sequences, right. So, this during preparation of this library, this next era transposes adapter was used. So, this appears in the read data, right. So, we have discussed why adapter contamination happens, what is the issue. This happens when the fragments generated from the genomes are small, right, and the sequencing process will go into the adapter sequence. Okay. So, once you have this adapter sequence, right, this means we need to remove adapters from the data. Okay. So, we need to do something called trimming. Okay. We have again discussed what is trimming, etc. Okay. So, this is for the read 1. Let us look at the read 2 data, right, what are the results. We will again have very similar summary file, and again you see two points where you see this fail. Okay. So, you have part based sequence content and then you have the adapter content. Okay. And we can again without blindly believing the statistic, we can actually go and look into the images. Okay. So, part based sequence content, we again see very similar thing, right. In the beginning you have these fluctuations and that is expected in case of Illumina, but for the rest of them it is quite normal, and this is an AT biased genome. So, it looks okay. So, we are not too worried about this warning here. Okay. But then we also need to check the adapter content, right, and here again we see very similar pattern, right. We see again we have many reads that contain adapter sequences, okay, and the similar observation we have this next era transposes sequence, right. So, this kind of helps us in evaluating the data, right. So, the data quality score wise looks great, right. We have checked there is no warning, no issues, but there are few problems, right. One is the adapter content. Okay. So, this is something that we need to address before we actually go for any downstream analysis. Okay. And this is where the pre-processing step will come. Okay. So, let us now go into the pre-processing step. Okay. As we have mentioned, right, once you have done the quality control, you get the results, you evaluate the results, you go into the pre-processing if it is required. In our case it is required, right. We have adapter contamination, we will do trimming and then we trim it back, we check it back with the fast QC, right. So, once we have done trimming, we check it through the fast QC again. Okay. So, that let us do that, right. So, let us first come out of this folder, right, and let us do the trimming. Okay. So, for trimming we have discussed many tools, right. If you remember, we have discussed at least 3 tools we have mentioned. I have given you the link, but today we are going to use one tool, which is the BBDuck. Okay. So, BBDuck is a tool that utilizes some of this information about the adapters to remove this from the read data. Okay. So, we can actually set this up BBDuck, right. We can simply say BBmap. This is actually a package, right, that contains this function BBDuck. Okay. So, we can go into BBmap, right, and we can download and extract this BBmap. Okay. And if you can see on the left side, I will zoom in a bit. On the left side you can see you have this BBDuck, right. So, BBmap has a lot of functions, we will not go into all those, right. So, I will just simply look at this BBDuck because this is the function that is useful for us. Okay. So, you can see there is an explanation, a lot of details, how to use this tool, etc., what are the options, what are the command lines, what is the algorithm that this tool uses. Okay. So, you have DUK, which means decontamination using KMAS, right. So, if you have adapter contamination, you want to do trimming, you can use this tool. Okay. You can do also quality trimming or filtering, and you can do adapter trimming. You have all these options here. You can see this adapter trimming or contaminant filtering, right. So, we will do this adapter trimming using this tool. Now, what is the algorithm? How does it do this? We will not go into that, right. There is a lot of details. If you are interested, you can always read, right. You have all the instructions, also how you should run this tool with your read data. For example, this is what we are going to use bb.sh in1 equals to read1.fq and then in2 read.fq out1 the output file names, right. So, you give input file names if you are working with paired end data. In our case it is paired end data. So, we will utilize this command and then we have the output file names. Okay. So, you have to give these 2 output file names. Now, there are a lot of parameters, right, in this algorithm. You can also see the memory usage. You can actually also control how much memory this tool will use, okay, because if you are working on a system that does not have too much memory, you want to use this flag. Okay. And then you have lot of these parameters you can see. Okay. Again, without going into the algorithm, right, these parameters are part of the tool, and sometimes you need to assign certain values for getting the best results. Okay. And this is something that you have to kind of do trial and error, right. You have to optimize these parameter values to through trial and error. You kind of set certain value and see whether the trimming has occurred properly or not, right, and then again go back, modify. If it has not happened, you go back and modify the parameters and try again. Okay. So, there are some parameters that work pretty well, you can probably find out once you have tried this out. Okay. So, what we will do is we will actually run this, right, through BBDuck, right, our data, because we have adapter contamination, and you will see these commands. Okay. Now, I will explain this command, right, what you have in here. Okay. So, this is the function, this is the command that will remove the adapters from your data. We have this in data, in 1, in 2, because we have paired end data. We will use out 1, out 2, because we are dealing with paired end data. So, we will get 2 output files. Then you have something called ref equals to adapters.fa. Okay. So, this actually refers to the adapters file that is present within this folder. Okay. So, BBmap has its own adapter sequences stored. So, this contains list of all adapters that are utilized by different platforms. Okay. So, these adapters can be then removed by this tool. Okay. What this tool will do, it will match, right, these sequences of the reads against these adapters, and whenever it finds some match, it will remove those adapter sequences. Okay. And then you have certain parameters. Okay. Again, we are not going into these parameter values, how this algorithm works, but these are the parameters that they have mentioned we can use, and you can also change this in certain sequences. So, there is a very nice explanation following this, okay, how we actually remove these adapters. And if you are interested in quality trimming, then you have to follow certain other parameters, etc., and we will not go into that because our quality score is pretty good. Okay. So, let us actually run this, right, this through our command line. Okay. And so, what I have done is I have actually optimized these parameters, and I have stored these parameter values in this file. Okay. So, the first step is setting up this BBmap. Okay. Now, you can see you can download this BBmap from here, right. You have this BBmap download here. Okay. You can download here this tar file, and you need to set this up. Okay. So, what I have done is I have downloaded this already in my system. You can see this here, this BBmap file. Okay. And we need to extract this, and we need to utilize this too. Okay. So, to extract, this is a tar.gz file, right. You remember hopefully this completion that we have talked about, and that information will be very useful. This is a tar completion, tar archive, right, with gz completion. Okay. So, we need to use this command tar minus xvf or zxvf, right, and BBmap. Okay. So, this will decompress the whole thing. Okay. So, once we do that, right, it is extracting all the functions, all the tools. Okay. So, it is done now, and we can check what has happened. Okay. You can see it has created a folder called BBmap, okay, inside which you have all the files, all the functions, all the tools. Okay. So, let us look at those functions, etc. You have lot of functions again, and as you have seen, BBmap can do a lot of things, but we are interested in only one function, right, which is the bbduck.sh. Okay. So, let us check if this function is there, right, bbduck.sh. We can also check, right, we can also check if this has permission, right, for execution. Again, if we download, sometimes there is no execution permission, but in this case we have, right. If in case you do not have this execution permission, you can change the permission using chmod. Okay. So, here is the function that we will use. Okay. So, it is download, it is very easy to set up, right. You just simply download the tar.gz file, you extract, and you have the function. You do not need to do anything for this program. Okay. So, as I was saying, we have tested different parameter values for this data, and we have optimized, right, which parameters actually work the best for these files. Okay. So, let us see, and I have stored this in this file here. Let us look at this, right. So, here is this, I have stored in something called bb.1.sh, just to distinguish from the bb.sh, right, and it actually stores the full command that we will be using. Okay. Right. So, if you are working on the command line, you can run this as a script itself, but if you want to just run in the terminal, you can copy this command and run in the terminal. Okay. So, this is what we will do. Okay. So, we will copy this command and we will run in the terminal. Before we do that, I will explain what this command actually means. Okay. In the first part here, bbmap bb.sh, we are calling that program bbdoc.sh, okay, inside this bbmap folder. Then we have this minus xmx2g. Okay. So, it means it will limit the usage of RAM. Okay. So, it should not use more than 2 GB RAM, right. So, if you are working on a system where you do not have too much of memory, you want to limit this memory usage, right, otherwise it might crash the system. So, this is how it is done, minus xmx2g. Then you have the input file names, okay, in1 d12 17539 etc read1 part.fastq. This is the first fastq file, then you have the second fastq file, right. So, this is required because we are dealing with paired end data, right. So, we need to give both files at once. Then you have the output file names that we can specify, and we can specify anything that we like, right. So, I have mentioned iteration 1 read1 trim.fastq just to distinguish from the original file names, right. This is the read1 file, but this is also trimmed, and then also I have mentioned iteration 1 because we might need multiple iterations to get to the optimal parameter or optimal data quality, right. So, that is why I mentioned iteration 1. You have these two files, right, out2 also, this is because you have paired end data, right. If you are dealing with single end data, you do not need to mention this in2 or out2. Okay. Then you have to give the reference sequences, right. So, what are these reference sequences? These are the adapted sequences that bbdoc will use to remove those adapters from the read data, right. So, this contains again all the adapter sequences. Okay. So, bbmap resources adapters.fa, I will show you in a moment what this file, where this file is and how this file looks like. Then you have certain parameters that this bbdoc has mentioned. Okay. So, let us run this, right. Before we run, I also want to see this, right, this adapters.fa file. Okay. So, let us quit, right. You remember now the vi editor, okay, :q, we do not want to do any change, we want to just quit. Okay. And we want to check this file, it is in this directory vi resources adapters.fa, right. So, this is where the adapters.fa file is, this is in FASTA format, right, .fa means FASTA format. And here you are, right, you have these adapter sequences, you have reverse adapter, TrueSec, universal adapter. So, these are adapter sequences utilized by different platforms, right, Illumina as well as other platforms. Okay. And you can see this, and perhaps you can also search if you see this NextEra, right. The search function in vi, that comes really handy here, right. So, you can see slash, and the moment you type next, immediately you see this NextEra, right. So, you have this NextEra adapter sequences here, which means we can now use this, right, file to remove our adapter sequences. Now, why I am showing this adapter file? Why do you need to know about this file? Imagine you are using some adapter sequences that you have yourself designed. Okay. These are not commercially available adapters, these are the adapter sequences that you have designed yourself for your experiment for whatever reason. Now, these adapters.fa file will not contain those adapter sequences, right. So, in your data, if you see some sort of adapter contamination, right, you want to update this file to remove those adapter contamination. Okay. So, this is something where you have to add this. Okay. You can simply add this by vi, right. You can simply add a header sequence saying your adapter name, right, and then give the sequence and just save it with :wq, right. So, here we are not saving anything, we have not written anything, but in case you do modify this file, you can save this by :wq. Okay. So, we have now seen this adapters file and we can simply run this now. Okay. which will remove this adapter data hopefully, right. So, let us run this. So, I will simply copy this, right, and then I will paste here, okay. And we have explained, we have understood what each of these commands actually mean, okay. So, let us now enter, right. The moment I press enter, it will start this streaming and this is done, okay, and you will get certain statistics, okay. So, it will give you some statistics regarding how much memory it utilized, etc., in this system, and then it will give you some statistics about the reads, okay, that it has processed, okay. So, here it give you some input about the input, right. It has about 50,000 reads in total, right. It processed 25,000 plus 25,000 in 2 files, and it processed about 6.25 million bases, right. So, in this read data you have 6.25 million bases. It has streamed certain reads. So, it has streamed about 13,000 reads and about 676,000 bases, right, roughly, which is about 10% of the data, 10-11% of the data. It has also streamed other reads. These are 2 different ways it has streamed, right. So, 1600 reads, again 9,700 bases. It has not removed any read completely. So, total removed 0 reads. It has all the bases, right, and the result is it has all the reads, right, 50,000 reads. The stream data contains all of these, and the bases will be around 5.56 million. Okay. So, from 6.25 million we have now come down to 5.56 million, right. So, it means some sort of trimming has happened, okay. And then there are some speeds, how much time it has taken, etc., etc. Okay. So, once this is done, the next step is to actually run it through the quality control pipeline, right. We want to check quality again. Okay. This is important because we want to make sure that these adapter sequences have been removed, right. So, let us do that, and here we will get the stream data, right. So, we have named these iteration 1 read 1 trim.fastq and iteration 1 read 2 trimmed fastq, right. Now, again running the quality control is very easy, right. We have to run through fastqc, and we follow the same command as before, right. We are running fastqc. Here the input file names will be different, right. So, input file names will be the iteration 1 read 1 trim.fastq, right, and iteration 1 read 2 trimmed.fastq, right. These are the trimmed files which we want to check through the fastqc. Okay. So, let us run this, and again we get very similar kind of output, right. All right. So, this is quite quick because we are dealing with small data, right. We are not dealing with the full data. As you can imagine, if you are dealing with the full data, it will take more time. Okay. So, this is complete, right, and we have generated these fastqc files. Again, you can see these fastqc files here, read 1 trimmed fastqc.html and fastqc.zip. Okay. We follow the same process. We create a folder, right, where we say mkdir trim data, right. So, we can move all the trim data there, okay, and we can move this fastqc now again using this command mv, and we go there. To trim data, okay. We move inside trim data folder, okay, and we again unzip. We are following the same steps as before. We have done this for read 1, now we do it for read 2, okay, and we have generated these folders. Okay. So, we have this fastqc folder for two reads, right, 2 data sets, and then we can now go inside these folders and we can check the quality again. Okay. And the quality that we will be focusing on mostly is the adapter content, right. That is what that was the problem earlier, right. We had adapter contamination in both of them. Okay. So, let us go into that folder and see what actually has happened. Okay. All right. So, let us see where is the data. Yeah. So, here is the trim data, right, and we can go inside. and again let us look at the summary. Okay. Now, the summary says okay, everything is passed except per-base sequence content. Okay, we are not worried about that too much, but it now gives warning about sequence length distribution. Okay. So, this is something that is new, right, and if you think about it, this is expected, right. If you are trimming sequences, your sequence length distribution will change and some of the reads might be very, very small. Okay. So, let us go into the images, right, and this will tell us the full story, right. If we first look at adapter content, now it looks great, right. There is no adapter contamination here. Okay, everything is gone. Earlier you used to see this like increase in adapter content, but that is gone. So, that problem is resolved, but there is some other warning, right. Where is this sequence length distribution, and you can now realize why this warning comes, right, because you have certain reads that are very, very small. Okay. You have most of the reads are of length 125, right. Here you can see that this is the peak for this sequence length, but then you also now have some reads that are smaller, and especially here you have reads that are very, very small. Okay. So, this is why the warning is right. Okay. So, we can now look into the other one as well, right, which is for the read 2. Okay, and again we can look at the summary file first, and you see similar results. Again the warning for sequence length distribution, but the adapter contamination is gone. Okay, it's passed, there is no adapter contamination, and we can confirm this through these images again, right. So, here is the adapter content, right, and it looks great, and we also have the sequence length distribution, and it is very similar story compared to the other file, right. Here you have sequence length, right, where most of the reads are of length 125, but you have some reads that are very, very small, but that is okay. I think we can go ahead with this data and it looks fine. It does not need any further pre-processing. Okay, so I think coming back to the presentation, we have actually gone through these steps, right. We have done this data quality control, we have evaluated the data, we saw some problems, and then we did some pre-processing, and after pre-processing we have again run the quality control and we have evaluated again, right. So, to summarize what we have seen, so data pre-processing is an iterative step, right. So, it requires trimming and re-evaluation of quality, right, and whether it might require some other sorts of pre-processing, that is again dependent on the data that you are working with. And data trimming is required when there are concerns regarding read quality or adapter contamination. So, in our case it was adapter contamination. So, we went through pre-processing, data trimming, and then also verified that the trimming actually worked. Okay, I will stop here. Thank you.